Chapter 21 is the follow-up chapter to the heart. This is the chapter where we talk specifically about blood vessels and understand that the vessels are the tubes that carry blood, and so certain things in terms of physics will happen in those and with those tubes. And what I mean is vasoconstriction, vasodilation to shift is part of your homeostatic shift with regard to blood vessel pressure, response to viscosity of blood, and also volume of blood, among other things. The key players in vessels are arteries, arterioles, capillaries, venules, and veins. And arteries, their definition at this point, carry blood away from the heart. Arteries carry blood away from the heart. Arterioles are the smaller branches, so smaller branches of arteries. Capillaries are the smallest blood vessels, and capillaries are specifically the location between blood and interstitial fluid, and capillaries are the exchange of the gas exchange area. Venules collect blood from the capillaries. Venules collect blood from the capillaries, and veins return blood to the heart. And so you have, in this image, you have from the heart, you have arteries into arterioles branching off into capillaries. Capillaries, then that's where the interchange between tissues are. And then you have venules and then veins. You also see the difference in size of the lumen, or I mean of the vessel wall. These circles are not to scale. Arteries are much larger than capillaries, but the number and layers of tissue, it's a pretty good explanation of layers of tissue. You have an outer, an inner, and then a muscle layer, and we will learn more about those with regard to the walls. The walls and veins contain three distinct layers, the first being the tunica intima. That tunica intima is the innermost layer, and you can see it on the artery on the left. Tunica intima has the almost the purple zigzag line on it. It includes endothelial lining, endothelial cells, and a connective tissue layer. There's also an elastic membrane there, that internal elastic membrane that allows that that vessel to stretch. So allows for a little bit of stretch based on the amount of pressure and volume that's going through that particular vessel at that time. Then the tunica media, that's the middle layer. You have concentric sheets of smooth muscle that are interspersed in loose connective tissue. And what that does, having that loose connective tissue there, allows that muscle to bind to the inner and outer layers. So the smooth muscle is what's responsible for the vasoconstriction, vasodilation, but the layer needs to bind to the inner and the outer layer, so you have the connective tissue there also. And then the external, the external layer, the tunica externa, which goes by other names depending on where you are in the body. You also have the tunica adventitia, but the tunica externa is the outer layer, and that's connective tissue. It anchors the blood vessels to other tissue, so anchors the vessel to adjacent arteries or adjacent tissue. It's made up of collagen and elastin, but in veins, because we are comparing and contrasting here, in veins you have elastin fibers and smooth muscle fibers in the tunica externa. The very, very small vessels are called the vasa vasorum. Those are the small arteries and veins in the walls of large artery and veins, and they supply the cells of the tunica media and externa. These are living tissues. It supplies the nutrients that these tissues need to exist. Compare and contrast the differences between the two or the similarities. They tend to run side by side. Arteries tend to have much thicker walls because they need to withstand the high blood pressure. An artery has a small round lumen, and you can see that lumen in this image. And a vein has a large flat one, which basically kind of stays flat. It doesn't have much substance. By the time the blood gets to the vein, it is not under an extreme amount of pressure, so it doesn't need to withstand that pressure. Arteries have, the big difference between arteries and veins is veins have valves. Blood flows through in the body in the following order. First, the elastic arteries, elastic because they need to withstand the pressure of the blood coming through. This is blood coming right out of the heart. They also need to help move that blood along. That blood is moved out of the heart via the left ventricle, but these elastic arteries having this give can actually move it along the systemic circulation. So you have elastic arteries. They also are called conducting arteries. They're the larger vessels like the pulmonary trunk and the aorta. The tunica media is a little different, has many elastic fibers and just a few muscle cells. So a little different than what we just described. And those elastic fibers actually pulse the, help with the pressure, so even out the pulse of the blood coming out of the heart. And then after the elastic arteries, you have the muscular arteries. They have many muscle cells. And so you can think if you have many muscle cells, these are the arteries that can shift the amount of blood that's going to a particular area. We've talked a little bit about temperature control and how blood helps you control your core temperature, your body temperature. These would be the arteries that could actually diminish blood in an area or increase blood to an area. And then you have arterioles. They have little or no tunica externa, and they have a very thin tunica media. The capillary, then, we will talk about it a little bit more in a follow-up slide, but they're smallest, and as I said, this is where gas exchange takes place. Capillaries are two basic types. You have continuous and fenestrated. We have a better picture of them. The continuous are continuous epithelial cells. Fenestrated actually has these pores that allow certain things to leave, allow more things to exit the capillary bed than the endothelial cells. From capillaries, then you go on to venules. And with regard to venules, they have little externa and then endothelium. On to medium-sized veins where you're having a little bit of the media, a little bit of each layers, and as you can see, as you move on up from medium-sized to large veins, very similar in structure. Capillary bed networks, the things that happen in the capillary bed, and this is one image. The blood flows through the capillary beds, but it's regulated by sphincters. So you have the ability to shunt blood in a certain direction to a certain area. This would be in response to local chemicals. Say, for example, if you have an inflammation in in a particular area, have an injury in a particular area, there may be a reason why blood would be shunted to that area as opposed to another one, very locally. We're not talking globally, but very locally. And these sphincters can do this. This is where there's interchange between interstitial fluid, that's the fluid outside of the of the circulatory system and the blood itself. Items get out of blood and into blood in various different processes. It can be active or passive transport. So active transport is going to require ATP. Anytime it's active transport, it's going to require ATP. Passive transport can be osmosis, so it can be solutes through that selectively permeable membrane, or water, basically evening out solutes, diffusion, filtration, and facilitated transport. So any of those processes can be the vehicle with which nutrients and substances get out of the blood into the tissue space, and also with which substances get out of the tissue space into blood. And the two types of capillaries, you have them here. You have continuous and fenestrated. Continuous capillaries have a complete endothelial lining. They are found in all tissues except epithelial and cartilage. And the function of continuous capillaries is basically to allow diffusion of very small things, but to block larger plasma proteins. So the smaller things can get out because you still have the junctions that will allow small things to get out of the capillaries, but the large plasma proteins, which you want to keep in the blood, like albumin, will stay in continuous capillaries. In terms of fenestrated capillaries, they do have pores. You can see the pores in this image, and they permit an exchange of water and large solutes between plasma and interstitial fluid. And you find these in endocrine organs, kidneys, intestinal tract. Any place where it's important that these large particles can get out, that's where you find these fenestrated capillary beds. If you have flattened fenestrated, you have sinusoids. Sinusoids permit free exchange of large proteins, and you have phagocytic cells monitor blood at sinusoids. So it actually is kind of, you're letting these large things out so they can be captured by these phagocytic cells, which is kind of interesting. In terms of veins, a little bit more about them. I know we've talked about them, but I did want to bring this up to show you how the valves work. So remember, in veins you have venules, medium-sized, large. But venules and medium-sized veins contain valves. The venules, the larger veins, no valves. So think of the larger ones being the superior inferior vena cava. This is what we're talking about large ones without valves. But the way the valves work is when that muscle contracts. You can see that on the image at the bottom left. When that muscle is contracting, we think of a muscle belly. We can see it actually bulging and contracting on the outside. Well, it's also contracting on the inside. That contracts, squeezes the vein. That squeeze opens the valve and allows the blood to move on through the circulatory system. If you are stationary for too long, that blood will pool. It won't circulate. It's one of the reasons why exercise is extremely good for you, because it does pump the blood. The venous return, so the blood back to the heart, is assisted by this muscular compression, so the constriction of muscles plus the respiratory pump. We'll start this up on the next one.